Hello, good morning, good afternoon, everyone. Welcome to our AMP online workshop entitled Next Generation Sequencing in MPNs, Unlocking Diagnostic Precision and Prognostic Power. I'm Chris Allen, a Medical Affairs Manager with Thermo Fisher Scientific, and I'll be moderating today's webinar. So, just before introducing our speakers, I'd just like to go through some housekeeping items. So, whilst the slide deck for today's presentation won't actually be shared, the webinar is being recorded and there will be a free on-demand version being made available on the AMP website very soon. So, we highly encourage you to submit any questions that you have during the presentation via the Q&A box so that we can address them at the end of today's session. So now on to today's speakers. So firstly, I'm delighted to introduce Dr. Haneen Madani, consultant hematopathologist at Bart's NHS Trust in London, part of the Specialist Integrated Hematological Malignancy Diagnostic Service. I think I prefer SIHMDS. She specializes in integrated reporting of complex hematology cases from bone marrow and lymphoid samples. So, Dr. Madani, brings a multidisciplinary background across internal medicine, oncology, and histopathology, and holds membership of the Royal College of Physicians. Her experience also includes early phase clinical trials, and she is a fellow of the Royal College of Pathologists. And her research interests focus on myeloproliferative and lymphoproliferative disorders, particularly T-cell lymphomas. Our second speaker is Marianne Grantham, clinical scientist and head of cytogenetics and molecular hematology at Barts Health NHS Trust. Her team delivers comprehensive genomic testing for heme malignancies, including national level MRD testing for adult ALL. Marianne has over 20 years' experience in genomic diagnostics and is a diplomat of the Royal College of Pathologists in both cytogenetics and molecular pathology. She's particularly passionate about integrating genomics into clinical care and advancing education, and also serves as an honorary lecturer at Queen Mary University of London and University College London. So that's the speakers. Very much looking forward to today's presentation. So over to you, ladies. Thank you.
Thank you, Chris. So these are our disclosures and the learning objectives. And so today we're going to give you an overview and to show how genomics in myeloproliferative neoplasms is used across the entire patient's journey along diagnosis, risk stratification, and therapy selection. And so the classification of myeloproliferative neoplasms has evolved over the years. And currently there are two published classifications that we know of, the WHO and the International Consensus Classification. There are currently discussions regarding unifying these classifications. So hopefully soon, in the next couple of years, we will have one unified consensus. And luckily that in myeloproliferative neoplasms, there are not many differences between the WHO and the ICC in the classification of these myeloproliferative neoplasms. So there are various types of myeloproliferative neoplasms, and they are clonal diseases of proliferation of hematopoiesis, where there is overproduction of mature cells which produce too much of one or more cell types. And each subtype of the MPNs has a different biology with different clinical manifestations, different morphology, and a different genomic basis. I've also included here the myeloid lymphoid neoplasms with eosinophilia and tyrosine kinase gene fusions and mastocytosis, because these are also conditions that, with their underlying genetic defects, lead to proliferation in cell lineages. So both updated classifications refine the diagnostic criteria to differentiate disease subtypes, including the updated genomic relevance. And there are three myeloproliferative neoplasms, also known as the BCR-ABL negative myeloproliferative neoplasms here. And we have the polycythemia vera, ET, primary myelofibrosis, the early stage, the early prefibrotic stage, and the overt fibrotic stage. So they are all due to a constitutive activation of the JAK-STAT pathway by driver mutations, which may be the same, but still each has a different biology and a different clinical outcome. And they are diagnosed by fulfilling the criteria that requires incorporation of the clinical findings, the bone marrow morphology, and the genomics. And so these charts show the distribution of different mutation types in MPNs from the New England Journal of Medicine from a few years ago. It shows that PV almost exclusively has a mutation in the JAK2 V617F and more rarely a JAK2 exon 12 mutation. In contrast, the ET and PMF commonly have a JAK2 mutation, but there's a substantial subset that has a CALR exon 9 mutation, and a small subset have an MPL mutation. And in case of the ET, 10 to 15% are triple negative and have neither. So some cohort studies have identified that the median overall survival for ET, PV, and PMF and the poorest overall survival was seen in the pre-PMF and the PMF at 8 and 6 years, with better survival seen in ET and in PV. And vascular events are the main causes of death in patients with ET and PV, whereas the consequences of AML transformation are the most fatal events for patients with PMF. And to assess for these risks and determine prognosis, the factors examined are clinical factors such as age, symptoms, history of thrombosis, previous therapy, and also the biological factors, and these are mainly the blood counts as well, and as well as some underlying genomics as well. And all this information is incorporated to consider appropriate therapy such as anticoagulation and cytoreduction in the at-risk patients. Also, targeted therapies such as the JAK inhibitors may be beneficial in some patients. And most importantly, it's important to assess for patients requiring transplantation, especially in the PMF group who have the poorest overall survival. Now here the clinical, this diagram shows adapted from the Pasquer review, and it shows the clinical, biological, and molecular factors implicated in reduced overall survival in ET, PV, and MF. And that's shown in the top figure. In particular, MF has a clearly defined high molecular risk signature. And ET, for instance, the JAK2 V617F mutation combined with TET2 or DNMT3 are associated with a higher risk of thrombosis. And these have been used in the arterial and venous thrombosis scoring systems such as ARTS and VETS. And this helps in deciding on the therapy if these patients are going to require anticoagulation or not. In PV, a JAK2 V617F mutation with a VAF more than 50%, and in some other studies, it's been given a cutoff of more than 75% VAF, was proposed to be associated also with increased thrombotic risk. And now this is a similar figure showing the risk factors associated with fibrotic transformation and leukemic transformation. And this highlights the importance of not only the JAK2, CALR, and MPL mutation status, but also the broader genomic profile in determining these outcomes. So in this paper from the New England Journal of Medicine from 2018, the authors used a data-driven clustering method to subgroup MPN patients into 8 distinct genomic subgroups based on combinations of mutations and chromosomal abnormalities. And these subgroups showed clear differences in phenotype, including the proportions of ET, PV, and myelofibrosis, as well as clinical outcomes and risk of leukemic transformation. Again, highlighting the value of integrated genomic profiling for refined diagnosis and prognostication. So initially, the scoring systems were based on clinical factors, but over time, the addition of genomic data has refined these scoring systems, improving predictions. And currently, the current one, the MIPS 70 plus version 2.0, has incorporated the most comprehensive genomic data so far. And in PV and ET, the management priorities predicting and preventing fatal vascular events. And in MF, the absolute priority is accurately predicting overall survival to justify high-risk patients for hematopoietic stem cell transplantation. So this is a very nice visual abstract from the Pasteur paper, and it summarizes all the risk classifications and the different scoring tools that were discussed. And risk stratification is no longer a monolith. Precise MPN management requires isolating the specific clinical outcome and applying the targeted clinical molecular tool for better management of these patients. And for chronic myeloid leukemia, this is the BCR-ABL myeloproliferative neoplasm. We could dedicate an entire webinar on this and discuss CML, but we just wanted to highlight that the importance that genomics plays across the whole patient pathway here, from the diagnosis through to treatment monitoring and risk stratification. And there are also now identified new molecular risk factors, such as the ASXL1 and RUNX1 mutations that carry strong prognostic and disease trajectory significance, particularly in advanced or evolving disease. So the myeloid lymphoid neoplasms with eosinophilia and tyrosine kinase fusions are not technically included in the MPN group of diseases in the WHO and ICC. But these diseases are driven by gene rearrangements that lead to cell proliferation. And they can present in a plethora of clinical and morphological manifestations, making their diagnosis quite challenging. But the most important thing is that in the first two ones, which is the PDGFRA and the PDGFRB rearranged subtypes, dramatically respond to TKIs. And now I will hand over to Marianne to tell us more about this exciting subject.
Thanks so much, Aline. So I'm going to talk a little bit today about some of the workflows that we used to use in our department and how we've changed them over time as our knowledge has increased and our understanding of genomics in the MPNs has changed. So prior to 2019, we used to test our myeloproliferative neoplasms sequentially for the canonical molecular mutations in MPN. And what I've tried to do here is I've actually adapted our workflow to kind of align from the AMP molecular in my pocket guide, just really to highlight the similarities. So samples received from patients with clinical suspicions of an MPN would have initially PCR testing for the JAK2 V617F mutation. And this commercial PCR assay that we used had an analytical sensitivity of approximately a 2% allele frequency. So for patients with a suspected diagnosis of PV, if we were negative for the V617F, we would then send the sample away to another organization for exclusion of the JAK2 exon 12 mutation, and that would be via high-resolution MELT technology. For patients that were referred with a suspicion of ET or PMF, who were negative for the V617F, we would then reflex to look for CALR exon 9 mutations via capillary electrophoresis. And if this was negative, we would then reflex again for mutation analysis of the canonical W515K and L MPL mutations. And for our patients with a suspected diagnosis of CML or just overarching MPN, we would perform, and we do still perform, a PCR procedure to look for the BCR-ABL fusion transcript. Now, there were certainly some limitations from this approach. You can see quite clearly how the number of different workflows and methodologies we're using, so it wasn't necessarily the most streamlined or efficient use of not only our equipment, but our workforce. It also had some diagnostic limitations, so we know that best practice for looking for JAK2 V617F, we ideally want an analytical sensitivity of down to 1% as a minimum. We also had fragmented logistics. So we were sending samples away, we were having to reintegrate reports coming back. This really wasn't efficient. Also, our PCR for BCR-ABL was only looking for the most common fusion transcripts. So there were a potential of patients that had rare fusion transcripts that we weren't picking up, and we were having to send those away to another institution. And we actually had a couple of cases where patients had polymorphisms in the JAK2 gene, which meant that the primers used in our commercial assay for the V617F gave a false negative result because the primers didn't bind. And so when they were subsequently sequenced by NGS in the future, we actually revealed that the patients did have V617F mutations, but that was masked by the previous PCR assay. And so Haneen has already touched upon this kind of profound paper from New England Journal of Medicine from 2018, when Grunfeld and team reported the genetic analysis of over 2,000 patients with an MPN. And they identified these 8 genomic subgroups with these distinct clinical phenotypes with the risk of leukemic transformation and event-free survival. And their comprehensive genomic characterization provided a classification of MPN that was driven by 18 key genetic aberrations, including epigenetic regulators, splicing factors, and signaling pathway genes. And I think their study demonstrated quite nicely the clinical value of comprehensive genomic profiling compared to limited testing just for those canonical driver mutations in JAK2, CALR, and MPR. And so it was around this time that we began to review our own workflows and to see if we could be more efficient and potentially yield more clinically useful data by incorporating our MPN samples into our NGS service. So when we were thinking about NGS testing in MPNs, we wanted to really think about, well, what do we want from a panel? We want to make sure that it's comprehensive so that it has the possibility of having both DNA and fusion targets, particularly thinking about those eosinophilias and those rare transcripts in CML. And also, it needs to be able to pick up those non-canonical mutations seen in JAK2 and MPN. We'd quite like to have the technology to provide rapid results, so between 24 and 72 hours for those very clinically unwell patients. We wanted any panel that we selected to be reliable and accurate, so ensure that it could detect variants in those hard to sequence regions. And we needed to make sure we had confidence in the content of any panel. And as I've already mentioned, we really wanted to think about analytical sensitivity. So could we really deliver that 1% allele frequency in JAK2 V617F? And I think what's always really nice is there any scope for custom design options? It's always nice to have the opportunity to update panel designs when our knowledge increases. So at the time that we streamlined our workflow, this is the panel content that we first transitioned our MPNs onto. And so we opted for an amplicon-based panel, which did in fact incorporate both DNA and RNA targets. And so here I've now updated our workflow using NGS to replace those individual PCR steps. And so we still use a PCR-based approach for our CMLs at initial screening, but we do regularly use the RNA panel to screen for rare transcripts and to be able to offer rapid fusion detection in referrals for eosinophilia. We also extensively validated the sensitivity of the panel so that we could pick up those JAK2 V617Fs down to at least a 1% allele frequency compared to our previous PCR. And it has also really helped streamline our workflows in the laboratory. So we use the same single workflow for all our myeloid malignancies. And so here I've summarized some of our NGS results dating back from 2023 to the beginning of 2026. So this now totals almost 7,000 samples. And I think as expected, we can see that the JAK2 V617F mutation is the most frequently observed finding. And we've got a really small number of exon 12, JAK2 exon 12 mutations and non-canonical JAK2 mutations that we can see just sitting at the top of that JAK2 bar on the graph. Next, we're also seeing a high proportion of mutations in the epigenetic modifying genes, the TET2, the DNMT3A, and the ASXL1, which is unsurprising, really, as they frequent in both MPNs, but also, as we know, seen commonly in clonal hematopoiesis of undetermined significance. But I also think this graph nicely highlights the additional genomic data that would have gone undetected using our previous PCR workflows. As we look at the data in a little more detail, we can see that 73% of our referrals didn't actually have a mutation. And this may reflect the clinical practice to send for genomic testing prior to excluding all reactive and secondary causes of a raised blood count. But we can see that of the 27% of samples with a mutation, 17% had a single mutation, but 10% had multiple mutations. And whilst JAK2 was the most commonly mutated gene at 30% of all samples, 70% of samples showed mutations in other genes. So I think the key finding here is really that NGS does provide a broader mutation detection and more clinically actionable data than when we were just doing PCR on its own. And just to have a little bit of a deeper dive into that data that I've presented, we found, and this is again not surprising, that the most frequent mutations were affecting the JAK-STAT pathway. And then, as I've mentioned, followed up with the epigenetic modifiers. But crucially, that mutations are also involved in the spliceosome genes, the RAS-MAPK pathways, and other cellular processes, including the tumor suppressor genes. And so I think this diagram just kind of nicely summarizes the key molecular pathways that drive MPNs. At the top, you've got your primary driver mutations, your JAK2, CALR, and MPL, which are typically mutually exclusive, although not always, and lead to that constant cytokine-independent activation of signaling. These mutations converge on a central signaling hub, the JAK-STAT pathway. And activation here drives gene transcription that promotes myeloid cell growth and survival. But we know that supporting pathways also contribute. The RAS-MAPK pathway enhances proliferation and differentiation, while the PI3K-AKT-mTOR pathway promotes cell survival and metabolic activity. And then we've got additional modifiers like the epigenetic regulators, TET2, ASXL1, DNMT3A, and the spliceosome genes, SRSF2, U2AF1, that can influence disease severity. And then ultimately, these pathways lead to increased myeloid gene expression, reduced apoptosis, and clonal expansion. And in some high-risk cases, loss of the tumor suppressor gene P53 can drive transformation to acute leukemia.
And so, I mean, the most important thing is that this genomic results cannot be interpreted in isolation. And that's why an integrated SRHMDS model is very essential to come with the proper diagnosis and the proper management required for a patient. Because, for example, in the non-BCR-able MPNs, a JAK2, all three of them, an ET and a PV and a PMF, they each could have a JAK2 V617F mutation, but they are different. So we have to incorporate the clinical information and the morphology and then the genomics to be able to come up with the proper the proper diagnosis and the proper clinical management path.
Okay, so we're now going to talk you through some interesting cases that we've had in our organization that we find useful to really demonstrate the benefit and the value of more comprehensive genomics. So the first case we're going to talk to you today is about a 53-year-old male. He was a beta delta thalassemia carrier, and he presented with severe anemia and splenomegaly. He reported clinically shortness of breath, fatigue, and weight loss. He had a raised white blood cell count, reduced hemoglobin, reduced platelets, and a slight neutrophilia. And he also had an abnormal blood film, which showed background red cell changes with nucleated red cells, occasionally hypogranular neutrophils, and pleomorphic lymphocytes. And perhaps of interest, there was no abnormal population detected by immunophenotype.
And so we had, this was the aspirate, unfortunately, was a tri-tab, as Marianne said. So this was the trophine biopsy. And as you can see here on the left box, you'll see the left picture, you will see that there's a lot of pink there. And there's a lot of small cells, smaller cells there, which are very dark, and they have lots of cytoplasm, very pink. And these are the megakaryocytes, and they look abnormal. A lot of them are, megakaryocytes should be hyperlobated, and you should be able to see through the nuclear chromatin. But here they're very dark, the nuclei are very dark, and they have irregular outlines, and they're not lobulated as they're supposed to. And at the background of these cells, you'll see that there are some scattered hematopoietic tissue. Some of them are erythroid, some of them are myeloid, but they are very reduced. So it is a hypocellular marrow, and it was also quite fibrotic. And on the right-hand side, you'll see the reticulin stain. And the reticulin stain shows all these fibers, all these black stripes. These are the reticulin fibers indicating background fibrosis.
And so a sample was referred for genomic testing. So we performed cytogenetics coupled with a limited FISH panel. The chromosomes, they're not very pretty, they're not very good quality. Not surprisingly, the sample was quite hypercellular. So the patient had a normal karyotype, and we didn't find any rearrangements or high risk factors with our FISH, although we did show 3 copies of the MECOM gene probe that's located on the long arm of chromosome 3. And then we performed our myeloid NGS panel, and we were able to demonstrate that the patient had the canonical JAK2 V617F variant at 11% allele frequency. And the patient also had the Q157P mutation within U2AF1, and this was at an allele frequency of 37%. And we ran the RNA fusion panel on this patient, and there were no fusions detected.
And according to the ICC, this patient meets the criteria for primary myelofibrosis and overt fibrotic stage. And we've inserted his information into different scoring systems. There's the GIPS and the MIPS 70 and 70 plus. So I just wanted to highlight here that these that the 2 different scores, even though the GIPS uses some high-risk mutations, the MIP70 uses a broader range of high-risk mutations. And there is a difference in the median overall survival. And in here, the overall survival does not look a lot, because in the GIPS, the overall survival is 2 years, and it's 1.8 for the MIPS. But in this scenario, yes. But a lot of these, we have seen quite a lot of big differences between these different scoring systems, and the fact that when the more high-risk mutations are inserted or included in a score, that it has a more accurate prediction of the patient's survival.
And so the current status of the patient is in fact that they were transfusion dependent, and they've been started on momelotinib and EPO, and stem cell donor search has been undertaken. So momelotinib is an oral kinase inhibitor that has activity against JAK1, JAK2, and ACVR1, and is used in the management of myelofibrosis. And it works through a dual mechanism. So firstly, it inhibits JAK1 and JAK2 signaling, and these pathways are overactive in myelofibrosis and drive inflammation and abnormal blood cell production. And by blocking them, the momelotinib reduces cytokine signaling, which helps improve the systemic symptoms like the fatigue, the night sweats, and the weight loss. But also, secondly, and uniquely, it inhibits ACVR1, and this reduces levels of hepcidin, a hormone that limits iron availability. So lower hepcidin improves iron mobilization, supporting red blood cell production and helping to correct for the severe anemia. And together, these effects not only control disease-related inflammation, but also address this anemia, leading to improvements in both constitutional symptoms and also, hopefully, transfusion dependence. So moving on to our second case. This is a 68-year-old male who presented earlier this year with a marked anemia and leukocytosis, and also a moderate thrombocytopenia. And again, like in the previous case, they had no abnormal population detected by immunophenotyping.
And I mean, very, very extremely high white count. And the morphology reveals the same. So the blood film showed a very increased neutrophils, and the aspirate also showed there's an increased, and it was very hypercellular and showed increased myeloid cells. Most of them are maturing into neutrophils. There was no blast excess. And I think at that time they thought it was probably a borderline dysplasia seen in the myeloid expansion. But on the Trifine histology, it was very hypercellular, and there was some dysplasia seen there, but mostly in the erythroid and megakaryocytic lineages, but not in the myeloid lineages. And there was numerous neutrophils seen there. So you could see here in the bottom two, this is the routine H&E stain of the Trifine biopsy, and you'll see that it's very cellular. These the white circles, these are the adipose tissue cells, and these usually you will have with this age about 50 to 60% cellularity at most, but here this is going over 80 to 90% cellularity. And you can see that all these there's loads of neutrophils, or these small hypersegmented myeloid cells are the neutrophils. And you'll see in the background you have some megakaryocytes. Some of them are these are the larger cells. Some of them are a little bit hypolobated, as we saw before. So it does kind of make you wonder if there's dysplasia or not. But there wasn't definite dysplasia that we can say really. So that's why the genomic investigations were very important, and we were waiting with bated breath for the myeloid gene panel.
So the sample was referred for genomic testing. There was a query around whether this person initially had CML, and we performed FISH for BCR-ABL, and that was negative. We also performed our NGS panel, and very nicely it showed 4 strongly clinically significant mutations in SRSF2, ASXL1, SETBP1, and CS3FR. So on the basis of those NGS results, the diagnosis of chronic neutrophilic leukemia was made. And so what we know here is with this disease, it has a variable prognosis, and it can be latent to highly aggressive. So it can be from 20 years down to overall survival of 6 months. And so for our patient, they were quite unwell, so they underwent cytoreduction and weekly transfusions. But what we do know is that, like in our patient, ASXL1 mutations have been associated with a poor prognosis. So moving on to our third case, patient C. This is a very recent case to come through our institution, so very, very timely. This is a case of a 41-year-old male, known HIV positive, who presented from a regional hospital. He showed high eosinophils and bone lesions. So the sample came in with a referral of query lymphoproliferative disorder or perhaps MPN. We can see he had a raised white cell count and a very high eosinophilia count. The immunophenotyping showed no obvious expansion of clonal lymphoid cells and no excess of CD34 positive or 171 positive progenitor cells.
And then here we have the histology from the Trifine, and there were many atypical cells there with irregular nuclei. These cells were and the marrow was markedly hypercellular, and there was a marked myeloid expansion with increased eosinophils. So there was loads of eosinophils in the marrow to mirror what has been seen in the peripheral blood counts. And there was also a slight increase in mast cells. And there was also an increase in the CD34 positive blasts at about 10%, and lots of background necrosis. So this was a very, very sick marrow, which was very hypercellular, and with lots of eosinophils, and also an excess of blasts, making us quite concerned for this being some sort of an MDS with excess blasts, or it's an MDS with transforming, at the verge of transforming.
So the sample came through to our laboratory for genomic testing, and because of the query around the eosinophilia, we performed FISH for the FIP1L1 PDGFRA rearrangement. We used a triple color probe, blue, red, and green, looking for a deletion of the CHIK2 probe to bring together the FIP1L1 and PDGFRA. And actually, on the peripheral blood, and on first pass of the bone marrow, the FISH appeared normal. But at the same time, we performed NGS. And on the DNA panel, we didn't detect any clinically significant variants. However, using the RNA best fusion panel, we detected the FIP1L1 PDGFRA fusion transcript. But most surprisingly, we also detected a set NUP214 fusion transcript. And this RNA analysis was repeated on the bone marrow. And following these findings, we reviewed the FISH results. And actually, if you looked at possibly upwards of nearly 1,000 cells by FISH, you possibly could have found 1 or 2 cells that showed the deletion. to confirm the rearrangement, but it was so low level. In reality, in the diagnostic setting, there's no way you would have called it as abnormal. So really, the fusion panel testing really came into its own here and proved to be much more sensitive. And the fact that we've got these two fusions is really quite interesting, and potentially reflects that this patient does have the myeloid lymphoid neoplasm with PDGFRA rearrangement that was presenting almost in a blast phase with a second fusion present. Although that we can find no evidence in the literature of a patient currently with concurrent FIP1L1 PDGFRA with set NUP214, but we are aware of at least one case in the literature of a patient that had co-occurrent FIP1L1 PDGFRA with a DEC NUP214 rearrangement. And so at the moment, this patient is being treated with imatinib and is being carefully watched to see how they respond and whether they need to escalate their therapy. But one thing I really just want to touch upon here is like the real importance for looking for these rearrangements. We know that the FIP1L1 PDGFRA are variable breakpoints. And so there is no one technology that can guarantee to detect all the rearrangements irrespective of the breakpoint. So in our hands, we tend to run both the RNA sequencing but also the FISH, because there are cases where the FISH picks it up and RNA sequencing doesn't, and vice versa. So I would really encourage organizations and individuals to consider running both tests to ensure you don't miss any of these critical rearrangements, because the presence of this rearrangement can be so damaging to patients' organs, etc. And so if we move on finally to our last case of this webinar, it's a 25-year-old female that was referred to us from one of our regional hospitals who had shown a repeated raised platelet count from about 2022 onwards. It wasn't particularly high, but it fluctuated between just over 400 to 500, relatively normal white blood cell counts, and marginally raised hemoglobin. And again, no obvious abnormal population apparent by immunophenotype. Now, what we're going to do is we're going to take this a little bit in a different direction now. We're going to show you the genomics first and then review the bone marrow morphology and histology. So a sample came in from the bone marrow for this patient for cytogenetics and NGS, and we showed that she had a normal carrier type. But on NGS, we didn't detect any of the canonical driver mutations in JAK2, CALR, and MPL. But what we did detect is a loss of function variant in the SH2B3 gene at an allele frequency of 30%. So this was a frame shift truncating variant. And so what I want to really highlight in this really nice schematic from St. Jude's is we know that mutations, particularly loss of function mutations, tend to cluster within these functional domains, so the PH domain and the SH2 domain, and our patient loss of function variant is located within the SH2 domain of the SH2B3 gene. And so SH2B3 encodes the adaptor protein link, and this is a key negative regulator of cytokine signaling in hematopoietic cells. And so normally the link protein binds to JAK2 and associated receptors, and it kind of acts like a brake, attenuating downstream JAK-STAT signaling. And this provides a critical feedback mechanism to limit proliferation and maintain controlled hematopoiesis. However, in the context of MPNs, loss of function mutations in SH2B3 impair this inhibitory role. And as a result, there is a reduced negative regulation of JAK2 signaling, leading to an enhanced and sustained pathway activation. So kind of the brakes have come off. And this amplifies cytokine-driven proliferation and survival signals. And this contributes to clonal expansion of myeloprogenitors. So, for example, in our patient, that increased thrombocytosis. And functionally, this can complement other driver mutations such as JAK2V617F and exacerbate the MPN phenotypes in those patients. So overall, SH2B3 loss of function removes that important regulatory brake on signaling, and it promotes dysregulated hematopoiesis, which is characteristic of MPN.
And so, I mean, after we have the genomics, this is the bone marrow biopsy from this young girl, and it shows on the routine stain here is seen on the right, on the left side, on the left side of the screen. You can see that the cellularity was about 80%, so that for her is a normal cellular marrow. The megakaryocytes were increased, and they showed mild atypical features, but not enough features to fulfill criteria for a myeloproliferative neoplasm. Some of these cells, megakaryocytes, as these are the very brightly pink ones with the dark nucleoli in the center. These ones, they were a bit hypolobated, and but overall there were some normal looking ones. And on the right side you'll see this is the CD61 stain, and that highlights those megakaryocytes. So the features were overall a little bit borderline, but none of the histological features were typical of a classical MPN. So it wasn't an ET, wasn't a PV, and was not a PMF. So the question was now with, is this now a clonal thrombocytosis of undetermined significance, which is a new term that has part of the new BSH guidelines to assist us in sorting out this problem with the thrombocytosis patients. And so, in summary, I hope that we have demonstrated how genomics is used across the whole entire patient pathway, and that it is important in the diagnosis, in the risk stratification, and in particular in refining the risk stratification and predicting the poorly prognostic factors, and also in therapy selection. So determining stem cell transplant, anticipating an JAK inhibitor response, selecting cytoreductive therapies, anticoagulant therapies, and guiding therapy in advanced disease. Thank you very much, and we're happy to take any questions.
Okay, thank you very much, Heneen and Marianne. So we have had quite a number of questions, so we'll do our best to get through as many as possible, but we will also be answering the questions offline after the webinar today. So one of the first questions came in was in multi-model detection. There are three platforms, RT-PCR and cytogenetics and FISH. So the question was, should we use all or one of them?
So the question was cytogenetics, FISH and PCR, or cyto, FISH and NGS? Sorry, just to
They
confirm.
said RT-PCR, cytogenetics and FISH, and they were wanting, should they use all or one of them?
Well, I think there's certainly a place for a multimodal approach, but for our MPNs, I would advocate using an NGS approach rather than a PCR approach, so that you can detect much more clinically useful data. When you're using PCR, you're only really going to look for those canonical variants. and there's going to be a lot of information that you can't see. In our organisation, in terms of cytogenetics, we don't perform routine karyotyping on MPNs other than in patients with myelofibrosis, so that we're looking for sort of adverse risk markers. So for anyone referred for ET or PV, we no longer provide cytogenetic testing because we believe that the added value there is quite limited. There might be exceptions where a patient has sort of a triple negative ET, they're a younger patient, and we feel like that we might be missing something. But I believe that we can get a lot of the information from sort of the RNA fusion panel, looking for rearrangements, and also for NGS. But I'm a true believer that not one method is going to replace everything else, and that it's really important that we retain the multi-model approach, because there are limitations with everything that we do.
Okay, thank you. So we have a question: to what extent can NGS be used as a primary diagnostic tool, particularly for BCR-ABL, JAK2, CALR, and MIPL, and blood versus bone marrow as a specimen source?
If I go for the sort of technology part first, and then maybe Haneen can talk about blood and bone marrow. We have so we use NGS up front now for our MPNs instead of sequential PCRs, because, as I've said, we found it to be much more efficient and cost effective, actually, because of the streamlined workflow, not needing to maintain so many assays to certain accredited standards, etc., and the extra information that we got. We have debated quite a bit locally here as to whether we should transfer our upfront screening tool for the BCR-ABL away from PCR and use the fusion panel, because our PCR, whilst it's multiplex, it can't pick up all the rare transcripts, whereas we know that this RNA fusion panel has picked up a significant number of rare fusion transcripts. So I think it really is a credible option. Certainly you can, when you use NGS, fusion testing doesn't take up much size on the panel, so you can multiplex up quite a large number of samples at once, and it is going to really ensure that you've got broad coverage of those fusion types. And maybe hand over to Haneen about whether we should be testing our MPNs on blood or bone marrow.
I mean, that's, you've answered the question really, but I mean, what I can add is that yes, the difference between only the peripheral blood is that we can get information much quicker, and it is non-invasive, of course. So I mean, and also it will be quite handy because in the new BSH guidelines for thrombocytosis patients, which are really hot off the press right now, and many of the patients, after going through their after going through filtering through the clinical investigations, and they have long-standing thrombocytosis, and many of them will have a peripheral blood sent to see if they have an MPN mutation, and if they don't have an MPN mutation, then most of these patients can't a lot of them do not require therapy, especially if they're not high risk. So it is I think it is something that we're going to be using more and more now, especially in these thrombocytosis patients. And so incidentally, it is non-invasive and it is quicker.
Thank you very much. Okay, so just to answer a kind of repeated question that's come in, which is asking for access to the slides and where will the recording be. So as anyone that's registered for the webinar will receive a link to the recording when it's available, and none of the slides will be made available but there will be the access to the recording. So I hope that answers those multiple questions. So back to the scientific clinical ones. So do you get many clinically unwell patients where rapid MPN screening is clinically beneficial? From MDT observations, these patients are typically treated as per hematology results first.
Yes, that's true. Many of them are seen in sort of outpatient clinics, but there are occasions, thinking about our case study of our patient with eosinophilia, he was exceptionally unwell and was experiencing significant organ damage from his high eosinophilia count. And so that rapid fusion testing was absolutely critical. And as I said, the FISH and the fusion testing were run side by side, and the FISH result came back as normal. But and if that had been the only test that had gone out at that time, the diagnosis would not have been made, and the patient would have been able to be started on the imatinib treatment. So the outcome might have been very different indeed. We do occasionally see patients that have had significant vascular events that have made them very unwell, and it's unclear whether that thrombotic event has been as a result of an MPN or some other process. And so we are asked for the results quite quickly. Also, again, we are asked to do quite rapid testing, certainly in a lot of females who have very high platelet counts in pregnancy, and it's wanted to be determined if there is an underlying MPN driving that raised platelet count or some other pathology. So not all of our MPN screens need to be performed rapidly, but certainly it's very useful to have the technology available that we can deliver that result in a very quick time frame.
Thank you. Okay. Sorry, I'm just sifting through the many, many questions. So how do you manage requests for repeat molecular testing in a previously negative patient? Do you have a recommendation, for example, a temporal gap between repeat testing, especially for JAK2 V617F?
Yeah, absolutely. And we do get that a lot, actually, which is how we picked up the patient that I mentioned that was negative by PCR because they had the polymorphism in the primer binding site that was where the primers were used for the commercial JAK2 assay. So we limit it to 12 months. We don't do repeat testing within 12 months for the standard screening this time. But in theory, we do repeat test if it's been greater than 12 months.
Okay, thank you. So a question, I think this has probably come from the UK. So, do you perform full myeloid panel screening for MPN referrals? I'm assuming the NGTD is the National Genomic Test Directory, and this is an English person, kind of NHS person. So the test directory has a dedicated MPN panel for JAK2, MIPL, CALR, and SH2B3. So it was just to clarify if you're using full myeloid panel.
Yeah, so we are using full myeloid for our patients because we run other myeloid malignancies through the same panel as well, and it just streamlines our workflow. But I am aware that there are other centres that use sort of smaller bespoke MPN panels. But I think you can see from the graphs that I showed in my talk the added value and clinical information. So if you use a much smaller MPN screening panel, you're not going to pick up that high molecular signature that's associated with MF, for example. And so then if a mutation is detected, you'd then have to go on potentially following a bone marrow or clinical review and run a repeat broader panel. And so from our perspective, we've done the cost analysis and can see that to do it the way we're doing it, it's the most cost efficient way for us.
Thank you. Okay. So another question, how to distinguish Jack2 chip from Jack2 MPN? So if there are clinical features of MPN, but not histological features, and a low VAF Jack2, is this Jack2 chip?
Good question. I don't know. Henning, do you want to take this one? You're on mute.
It's a very good question, that, and that goes back to why I said about the integrated kind of model, is that you have to look at the clinical features, you have to look at the histological features, and the genomics. So if you don't have the histological features, this patient cases like this need to be discussed in a multidisciplinary approach, by a multidisciplinary approach, and if they do, and then it will be decided if there are any if this is an MPN or not. So in situations like the thrombocytosis, if that is the clinical features that we have, we do have a new category, and that is the clonal thrombocytosis of undetermined significance. So that would be, that's in ET. But that is of course provided that you do not have any ET like histological features on the bone marrow biopsy. So yeah, I mean, I think the main thing is that you have to put all of the things together, and this needs to be discussed, and then decided if this will be called just a chip or just an MPN. And actually, the JAK2 does make up about 7 to 8% of chips. So it could be a chip.
Okay, thank you. So, unfortunately, it's time to wrap up. So, as I mentioned, any unanswered questions will follow up on afterwards. So, I'd like to extend a big thanks to our speakers, Haneen and Marianne, and to everyone for their attention today. And have a great rest of your day. Thank you.